Hello everyone. In this video lecture we will be discussing some questions related to the chapter Inherited Change for CIE 9700. So let's start. Sometimes a gene has more than two alleles termed multiple alleles. The ABO blood group system in humans is controlled by a gene with three alleles, IA, IB, and IO. Alleles IA and IB are codominant and IO is recessive to both. The blood group AB is the result of codominance. Explain what is meant by codominance. This question is for 3 marks, and to score 3 full marks, you can write codominance is when both the alleles express themselves in a heterozygous genotype. There are more than 2 possible phenotypes, and phenotypes of a heterozygote is different from either homozygote. Okay, to better to understand this, I've written down an example. For example, a flower color in a snapdragon plant is coded by a gene which has two alleles, CR, which codes for red flowers, and CW, which codes for white flowers. And in the exam paper, the examiner will give you this description because you cannot tell the phenotype obviously. So both the alleles are codominant and produce pink flowers when together in a genotype. And how can you judge or tell if codominance is given to you? Basically by the gene locus and allele. So the gene locus will be here and the allele will be the power to it, and IA as well if you've learned from last year. So we can write the possible genotypes and their respective possible phenotypes. So CR gives us red flower, CW gives us pink flower, which is codominance, and CW gives us white flower. So what did I say? What is codominance? It's when both the alleles express themselves in a heterozygous genotype. So you can see both the alleles are expressing themselves in a heterozygous genotype, and there are more than 2 possible phenotypes, as you can tell here as well, 1, 2, and 3. Not like dominance where you only have 2 phenotypes, and phenotypes of a heterozygote is different from either homozygote. Phenotype of a heterozygote is different from either homozygote. Pink flower is different from red and white flower. So if you write this in the exam, you can score full marks, and I have written this example so you can better understand. Also, codominance is always observed in a heterozygous genotype. And how many types of phenotypes are there in codominance? 3, like I have showed you. And in dominance, you only will find 2 phenotypes. If you have a gene with 2 alleles and its codominance, it's dominance only 2 phenotypes are possible. However, if there is codominance and you have a gene with 2 alleles available, you will get more than 2 phenotypes. Meiosis is one process that contributes to genetic variation. State precisely the stage of meiosis where single chromosomes line up on the equator. So since they're asking us precisely, so we should say that it happens during metaphase 2. That's where the single chromosomes line up on the equator, because during metaphase 2, the chromosomes line up along the equator of the spindle by the attachment of spindle fibers to the centromeres of chromosomes. I've drawn it here so you can better understand it. And meiosis 2 is just like mitosis. Outline the events taking place during anaphase 1 of meiosis. In anaphase 1 or during anaphase 1 of meiosis, the microtubules, they will become shorter or they will shorten. This will cause the homologous chromosomes to separate, and then they will move towards the opposite poles of the cell. Describe how crossing over during meiosis leads to genetic variation. Meiosis can lead to genetic variation because crossing over causes an exchange of allele between the gene loci of non-sisterchromatids of homologous chromosomes. When parts of non-sisterchromatids, they will break and rejoin to other chromatids during prophase 1, and this will result in a new combination of alleles. The lac operon is a section of DNA present in the genome of Escherichia coli or E. coli. The structural genes of the lac operon are only fully expressed when the bacteria are exposed to high lactose concentrations. Figure 6.1 is a diagram showing the lac operon and a nearby region of the E. coli genome. Figure 6.1 shows how the lac operon consists of structural genes and regulatory sequences. Use figure 6.1 to identify two structural genes. Complete table 6.1 to name each structural gene and its product. So they're asking to identify two structural genes. So you can say that is lac Z, lac Z, and lac Y. And lac Z or lac Z and lac Y produce beta-galactosidase and permease respectively. Gene 1 is transcribed all the time to produce its protein. This is constitutive expression. Explain why some genes show constitutive expression. Okay, basically constitutive expression is a type of gene expression which is always on. Okay, it's not like your regular gene expression. Constitutive gene expression means the gene is always getting expressed all the time in the cell. Like regular gene expression, it's a process when information from a strand of DNA or piece of DNA, it gets copied into RNA. This process is called or known as transcription. And then this information in that RNA is then used to make protein or a polypeptide. This process is known as or called as translation. So when a protein is being made from the information in DNA using RNA or mRNA, we say that that gene is being or getting expressed. However, some cells in our body, they're quite smart and efficient and clever. They don't like making proteins. Why? They don't like making proteins which are not required. Why? Because the process of gene expression, it requires a lot of energy and resources. That's why we have some cells. That's why we have cells which control which protein has to be made and when it has to be made. For example, insulin is a protein or a hormone which is secreted by the beta cells in islets of Langerhans from our pancreas, and other times insulin is not being made or produced. And the gene for making insulin is present in all the cells of our body, but insulin production only takes place in the pancreas. I repeat this point again. The gene for making or their codes for insulin production is present in all the cells of our body, but insulin production or insulin is only produced in the pancreas. So this variation or difference in gene expression occurs because there's a control switch that is already inbuilt within the cells, and in certain situations a gene has to be expressed all the time, and that condition is known as, or this process is known as, constitutive expression. For example, the genes which are involved in energy production from glucose, they always need to be expressed constitutively all the time, because we are living cells, and living cells, as long as they are alive, they're living, they will always require energy. So the answer to this question is, why some genes show constitutive expression? It is because proteins are always being needed all the time. Describe the effect of the product of gene I on the functioning of the lac operon. So basically, gene I goes for repressor protein. It will bind to the operator. So this binding will prevent the binding of RNA polymerase to the operator region, and because of this, transcription of lac Z or Z, Y, and A will not occur. If E. coli is put into a nutrient medium containing lactose, some new enzymes are synthesized. These are described as inducible enzymes. Explain what is meant by an inducible enzyme. So it's for 2 marks. For 2 marks, we can write the transcription of a gene coding for this enzyme can be induced by a substrate, also known as inducer. The structural genes of the lac operon are not expressed when lactose is absent. Suggest one reason why this is beneficial to E. coli. So, like I said previously, some genes are not expressed, or some cells they don't like making proteins because this gene expression or the process of gene expression, it requires a lot of energy and resources. So they asked for one reason, I've given you two. So like I said before, gene expression, it requires a lot of energy and resources. So the answer to this question is no energy will be wasted and no amino acids will be wasted, as some cells they don't like making proteins which are not required because the process of gene expression requires a lot of energy and resources. Figure 4.1 shows the biosynthetic pathway involving tyrosinase. Okay, so this is the pathway. Figure 4.1, there are a number of different mutations of the TYR gene that can result in an absence of melanin and cause OCA1A. These include a missense mutation caused by a base substitution is most common. A nonsense mutation caused by a base substitution is less common. An insertion mutation which is extremely rare. A missense mutation results in a complete polypeptide chain that does not fold properly to form the functioning enzyme. A nonsense mutation results in a shortened polypeptide. Explain why a missense mutation results in a different product from a nonsense mutation. Basically, in a missense mutation, we will get a totally different product as compared to nonsense mutation, because in missense substitution, an amino acid or one amino acid is replaced by another, resulting in the formation of an altered polypeptide. And in nonsense, or whereas in nonsense mutation, it leads to a base substitution resulting in a stop code. And to better understand this, I have drawn it here. It's like a rough diagram. So this is missense substitution and this is nonsense substitution. So as you can see, in missense substitution, one amino acid is replaced, resulting in the formation of an altered polypeptide. So you can see the T is replaced by an A in the second triplet, and this results in an altered polypeptide. And in nonsense substitution or nonsense mutation, you can see a stop code is made. You can answer this for 2 marks. The fruit fly Drosophila melanogaster feeds on sugars found in damaged fruits. A fly with normal features is called a wild type. It has a striped body and its wings are longer than its abdomen. There are mutant variations such as an ebony body, colored, or vestigial wings. These types of fly are shown in figure 7.1. Wild type features are coded for by dominant alleles A for wild type body meaning striped body, and B for wild type wings meaning long wings. Explain what is meant by the terms allele and dominant. So an allele is or alleles are different forms or different variants of the same gene. And a dominant allele is a type of allele which always expresses itself in both homozygous and heterozygous genotypes. 2 wild-type fruit flies were crossed. Each had alleles A and B and carried alleles for ebony body and vestigial wings. So these are recessive alleles. Draw a genetic diagram to show the possible offspring of this cross. So since they didn't show anything related to linkage, so we will follow independent assortment. So the tip and the trick to solve or to draw a genetic diagram as fast as possible, first you will write parental phenotypes, parental genotypes, gametes, genotypes and phenotypes of offspring, and phenotypic ratio. So since two wild type fruit flies are being crossed, so their phenotypes will be striped body and long wings for both, and their parental genotypes will be capital A, capital B, small b. It's the same for the other one. And their gametes will be capital A, small b, capital A, capital B, capital A, small b, small a, capital B, and small a, small b, and it will be the same for the other gametes. So I've already drawn the genotypes and phenotypes of, I've written them down, the genotypes and phenotypes of the offspring. So after doing all this, you can write the phenotypic ratio, which is striped body long wings, striped body vestigial wings, ebony body long wings, and ebony body vestigial wings. So striped body and long wings, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. Striped body vestigial wings are 1, 2, and 3. Ebony body and long wings are 1, 2, and 3, and ebony body and vestigial wings are only 1. So the ratio, phenotypic ratio is 9:3:3:1, and this is the expected ratio. When two heterozygous fruit flies in bee were crossed, 384 eggs hatched and developed into adult flies. A chi-squared test was carried out to test the significance of the differences between observed and expected results. So this is the formula. So chi-squared is the summation of observed value minus expected value, all of it squared, divided by expected value. Complete the missing values in table 7.1. So I've already taken out the values. So to find this value, we should do O minus E, which will result in 7. O minus E squared is 49, and O minus E squared divided by E is 0.680 quadruple 56. This will be shown in your calculator, but in the exam round it to 3 significant figure and you get 0.68. Calculate the value for chi-squared. To find the value of chi-squared, we will sum all these numbers in this row, which will be equal to 2.78. This is the chi-square value. Table 7.2 relates chi-square values to probability values. As 4 classes of data were counted, the number of degrees of freedom was 4 minus 1 equal to 3. Table 7.2 gives values of chi-square where there are 3 degrees of freedom. So as you can see from the table 7.2, as your chi-squared value is decreasing, your probability is increasing, and I've written here. And as your chi-squared value is increasing, your probability value is decreasing. Using your value for chi-squared in table 7.2, explain whether or not the observed results were significantly different from the expected results. So to answer this, you can use some tips and tricks which I have. First of all, our chi-square value, what was it? We just found out it was 2.78. We need to compare our chi-square value to the critical value. And how can you know what is your critical value? Your critical value is the value of chi-square at or when probability is equal to 0.05 or 5%. So we can see our chi-square value, which is 2.78, it is less than our critical value. And when your chi-square value is less than your critical value, the probability will be greater than 0.05 or 5%. And when probability is greater than 0.05 or 5%, there is no significant difference. And if there is any difference, that difference is just due to chance or by chance. So how can you remember this? I've written it down. So as you can see, when probability is less than 0.05, or we can say 5%, there is a significant difference. And when probability is greater than 0.05, there is no significant difference. And our chi-square value, when it is greater than our critical value, our probability will be less than 0.05 or 5%. And when chi-square value is less than critical value, our probability will be greater than 0.05 or 5%, which is in the case of this question. So you can remember chi-square value is inversely proportional to critical value. When chi-square value goes up, probability goes down. When chi-square value goes down, probability goes up. Is inversely proportional to each other. Chi-square value is inversely proportional to probability. So to answer this, you can say chi-square value is less than the critical value. So that means that the probability is greater than 0.05 or 5%, and this shows that there is no significant difference between expected and observed ratios, or observed and expected ratios. And if there is any difference, that difference is just due to chance, and null hypothesis is accepted. And null hypothesis is accepted because null hypothesis always says that there is no significant difference. I hope you found this video lecture helpful. If you did, please subscribe to my YouTube channel, and thank you so much for watching.